And this is going to be Chapter 3, Cellular Structure and Function, and this is going to be Part 1. Part 2 will also be found on the Blackboard site. This is about 36 slides. Introduction here. Just as atoms are building blocks of chemistry, cells are the building blocks of the human body, and that brings us to some terms here that we need to be aware of, which is cell theory. It includes 4 basic concepts. Cells are building blocks of all plants and animals. Cells are the smallest functional unit of life. Cells are produced through the division of pre-existing cells. So that means that they start off as cells, and what they essentially do is they'll divide and they'll make more cells. And each cell pretty much maintains homeostasis or balance. Now the cells are going to maintain balance for the entire body. So become very familiar with this term homeostasis, because you're going to see this again. Oops. Homeostasis. 1.0 here, studying cells, and this is an overview of the cellular anatomy. And we have more terms here. Studying cells. Cytology is the study of the structure and function of the cells. 2 most common methods of studying cells are under light microscopy or electron microscopy. Now what that is it blows up the picture of the cellular environment many times. Electron microscopes can actually almost look at the protein level or the DNA level whenever we're looking through an electron microscope. Whenever we continue on with this, the electron microscope, there's a couple different types that we'll kind of overview here. And under light microscopy, you have to have a pretty big bright source of light, and the more magnification you get, the brighter the light needs to be. Light microscopy, magnified to 1,000 times, is pretty much what you can magnify to. And then electron microscopy, TEM, transmission electron micrographs, which are photographs of very thin sections. So they kind of slice it, and it'll be a very thin section here that it'll blow up and shows like micrographs of this. Scanning electron micrographs, SEM, reveals a 3D image nature to the cell. So with an SEM electron microscope, we can actually see a 3D image or get a third dimension on the actual overview. And abbreviations here, you might see these some on the test. LM for light microscope, TEM for transmission electron micrographs, and SEM for scanning electron micrographs. And SEM is 3D, TEM is 2D, light microscope up to 1,000 times and by a light source only. Light microscope times 160 identifies the light microscope's magnification at 160 times. An overview of cellular anatomy here. We're going to talk about fluids in this section. Extracellular fluid surrounding the cell. So this is going to be everything outside the cell. So we have a little basic cell here. And when we talk about extracellular fluid, it's all of this area here. And then we have intracellular fluid, cytoplasm, and which is essentially inside the cell or the cell contents. So all of the stuff that's inside here. And then we have cytosol, which is actually the liquid, all the liquid that would be in this intracellular compartment, and the organelles. And we're going to see things in the organelles. Here, let me erase this here really quick. Make it a little bigger so everyone can see. The nucleus is an organelle. You have something in here that looks like a kidney bean, which is a mitochondria. You'll have the Golgi apparatus. You'll have the smooth and rough endoplasmic reticulum. So all the little parts that are inside the cell in this environment here are actually the organelles, and we're going to talk about those in detail. So here's the anatomy of a cell and kind of a picture. We'll kind of go through this. Up here, these finger-like projections are known as cilia. Now, cilia are pretty much for movement. So these things are only located on certain cells, and they allow the cell to move from one location to another. These microvilli here, these little area here, are a little different than the cilia. Microvilli if you have something that, if you're talking about surface area for absorption in this area here, the surface area for absorption, if it's flat, is a lot less than if we had a ton of these little microvilli here. Now the microvilli allow for more absorption to occur in all the little nooks and crannies. So an example of this is the lungs. And I'm going to erase all this so we can kind of see here. An example of this is the lungs. So inside your lungs you have two small organs whenever they're deflated that both together are about the size of a football, give or take. I can't even draw a football, but both of them together are about the size of a football, give or take. If you put them back to back on there, they'd be about the size of a football. Now, that's not very big, but if you actually took into account all the surface area that was inside the lungs at the cellular level from all the little microvilli, you would have an area the size of a tennis court. Now, that's a large area, and we're going to learn that later on whenever we talk about pulmonology. But that's what these microvilli do, is they allow us to pretty much apply more surface area to a very small spot. Alright, we have things like lysomes. These things are going to break down the cell, and we're going to talk about these secretory vessels. This is endo and exocytosis. This here, everyone will take a look at this, and this is figure 3.2 in your books. If everyone will take a look at this little section, this is endocytosis, is bringing fluid into the cell, and then let's see, let me see an exocytosis here. If it was an exocytosis, as it would bring it in and the body would do its thing to it intercellularly, if it had something to secrete, then it would secrete it out, and this is exo. So endo, exo. I'm going to erase this here again. Alright, and we have smooth endoplasmic reticulum here, some cytoskeleton formations, the actual cell membrane, which is a phospholipid bilayer, the Golgi apparatus. This thing is going to be responsible for both endo and exocytosis. Free ribosomes, which actually make proteins. We have the nucleus in here. These are all of our chromosomes that are located in this section here. Nuclear pores, where it releases the proteins out. Here we have rough endoplasmic reticulum, which the difference between smooth, which is this right here, and rough, which is this right here, is the rough endoplasmic reticulum has ribosomes on it. So all of these little dots are the ribosomes, and we see them over here as free ribosomes as well. This area here is the powerhouse, which is the mitochondria, and then we have a centriole here. We'll talk about those. Nuclear pores, fixed ribosomes, chromatin, which is the chromosomes, and so on. So all of these little parts inside of here are considered the organelles. This here, the fluid itself, is the cytosol. Cilia allow the cell to move, and microvilli allow for more surface area. The cell membrane, membrane structure and membrane transport. The outer boundary of the cell is formed by a cell membrane or plasma membrane. General functions of the cell membrane, and these are all, you need to look over these because this sounds like a pretty good test question. Physical isolation, or physical barrier, so you keep the outside out and the inside in. Regulation of exchange with the environment controls the entries of ions and nutrients. Well, if you lived in a little capsule, you would need both a supply of food and water coming in, and if you had any wastes or you needed any product out, you'd have to spit those out as well. Sensitivity. First part affected by changes in the extracellular fluid, which is considered the sensitivity. Structural support. Special connections between membranes and extracellular materials give a stable structure to things. So again here, I'll erase this up and kind of go over these. Physical isolation. regulation of exchange with the environment, its sensitivity, and the first part to be affected by changes in the extracellular fluid. So if you had a big change in the extracellular fluid, it would immediately start changing the intercellular environment because of the way that the intercellular environment interacted with its home and its surroundings. And then you have structural support. Special connections between the membranes and extracellular material give a stable structure. So, example of this, your heart is made up of a bunch of cells. Well, if they all wanted to do their own thing, you'd have a billion different parts moving in every other direction. None of it would be going the same way, none of it would be doing the same goal or anything. So these structural supports allow it to kind of isolate and keep within its own tissue type, so to speak. Membrane structure. The cell membrane is extremely thin and delicate. Ranges from about 6 to 10 nanometers, and that is tiny, 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 tiny. Not being able to see it by the naked eye, it can only be seen by microscope. You can actually see cells with an electron microscope. You just can't really see a good blow up of them where you could look at the detailed organelles inside the cell. Membrane lipids. We have every cell has a membrane, and it's a bilayer, phospholipid bilayer. It has a head on it and a tail on it. Head on it and a tail on it. Head on it and a tail on it. Head on it and a tail on it, and so on. All through the top and all through the bottom of this two bilayer. Now, hydrophilic and hydrophobic are the actual heads and tails of this. So if we'll look down here in this phospholipid bilayer, water-loving heads, so these heads on this phospholipid bilayer like water. The tails hate water and are inside, so the tails are the actual lipids. Have you ever got oil and water to mix? And the answer is no. But this phospholipid bilayer kind of does that. But it allows things to travel right across the cellular membrane if they're made of the right material. And we'll talk about that a little later. So hydrophilic, water-loving, which are the heads. Hydrophobic, water-hating, which are the tails, and those are the lipids. And then we have something called a phospholipid bilayer. Water-loving heads on the outside and water-hating tails on the inside. Hydrophobic tails will not associate with water or charged molecules. Pardon. Lipid-soluble molecules and compounds such as oxygen and carbon dioxide are able to cross the lipid portion of the cell membrane. So that means that oxygen and CO2 can travel right across this cell membrane because of this lipid tail. Alright, and here are the cell membranes. Now we have these protein channels that are scattered throughout the cell membrane, and these allow some things in and keep other things out, and we'll talk about that. You have gated protein channels, which are right here. You have just essentially protein channels, which are a gate that's open and they're only large enough to allow certain molecules or certain size molecules through it. You have these anchoring proteins here which present, and these proteins here will say this is how the body identifies it. I am me, I am self, I'm in the right spot. The gated protein channels would allow things like sugar in there, and we'll talk about that. The movement of glucose across the cell membrane. All of these little red dots are the heads, and all of these little tails are the actual lipids. So you have a phosphate head with a lipid tail. And the phosphate head is water loving, the lipid tail is water hating, or hydrophilic and hydrophobic. Membrane proteins. Several types of proteins are associated with the cell membrane. We have transmembrane proteins, proteins that span the entire width of the membrane. We have membrane protein functions. Membrane protein functions are receptor channels, carriers, enzymes, anchors, or identifiers. So whenever we start, and we'll talk about these in detail here, that They're either a receptor, so they're going to respond to something that's in the external environment. They are carriers, which will allow certain things in and not others. They are enzymes, which are easy money. So if you have enzymatic action, you don't require as much energy. You need part A and part B, and I can make part C. And then you have anchors, which will actually hold it on to other cellular products or identifiers. And the identifiers say, I am me, I am self, please do not attack, and so on. So as we go down through these, receptor proteins, sensitive to specific extracellular materials that bind to them and trigger a change in the cell's activity. So this guy's presenting, he gets interacted with, and now they're going to have another product come out of this, or the cell has been excited to do something. Channel proteins. Central pore or channel permits water, ions, and other solutes to bypass lipid portions of the cell membrane. So cell membrane here, and this protein channel would allow some things in, other things out, but only a certain portion. It's not gated though. It's free access to those parts. You have carrier proteins, binds and transports solutes across the cell membrane. Now, example of this. I have to draw two of these for this. An example of this is going to be glucose and insulin. Insulin works here, which allows this gate to open and allows glucose to enter into the cell. And since I messed up here, I'm going to erase all this here really quick. Alright, enzymes catalyze reactions in the extracellular fluid or within the cell. Again, an enzyme is easy profit. So I need part A and part B, and I can easily make C through enzymatic action, and it didn't cost hardly any energy. Anchoring proteins attach the cell membrane to other structures and stabilize its position. That way it can't run off and go do its own thing. Recognition or identifier proteins identify a cell as self or not self, normal or abnormal to the immune system. So again, if you have a cell here, and this is an actual identifier protein, if I was looking for a Y, but I actually got a G instead. If I was the immune system or a white blood cell, and I was looking for a Y, and this cell wasn't presenting a Y, it was presenting something completely different, I would say that that is not self, and I would target that for destruction. So that's how those identifier proteins keep your immune system from attacking itself. Diseases that we'll see that this gets messed up on, things like rheumatoid arthritis or any autoimmune disease, the cells themselves, there's either a malfunction of the white blood cells or there's a malfunction of the cellular environment where it's not presenting the information or the all-access pass like it should. Membrane carbohydrates. Carbohydrates form complex molecules with proteins and lipids on the outer surface of the membrane. Glycoproteins and glycolipids functions as cell lubricants and adhesives. Form a recognition system of the body and the immune system. So these glycoproteins, these glucose molecules attached to a protein molecule, actually make up, I am Roy Smith, I have an all-access pass to the system. It would just be like trying to get through security, and the system is continuously running. It's continuously looking at security throughout the system. Membrane transport. Permeability, which determines the substance, what substance can either enter or leave the cytoplasm. And then we have the term freely permeable, which crosses without any difficulty whatsoever. So a freely permeable fluid would actually go in and out of the cell freely, no gates, no nothing. Selectively permeable, allow free passage of some materials and stops others. So things like sodium or potassium, it may allow those to come in or out as it pleases, but it may stop things like proteins and sugars. passive processes, and this means without energy. So whenever we have a passive movement across the cell membrane, it required no energy, and it moved very freely from one location to the next. An active process, however, requires energy, and the best example of an active transport or an active process is going to be the sodium-potassium pump. On the cell membrane, and this is how muscles contract, you will have a pump that takes potassium outside of the cell and puts it in the extracellular area and takes sodium and runs it in. This movement or this change of electricity causes this cell to shorten, which in the big scheme of things would be the shortening of a muscle tissue or the movement of a muscle. Diffusion, net movement of molecules from an area of relatively higher concentration to an area of relatively lower concentration, and this depends on its perspective. A lot of people get confused whenever we start talking about diffusion and osmosis. Diffusion and osmosis have the same definition. The difference in them is osmosis is the diffusion of water. So again, diffusion goes from an area of higher concentration. So 3, 4, 5, 6, and 2. If we had a membrane here and I had 6 parts of X over here and 2 parts of X over here, to get this balanced, and this works just like a teeter-totter, how would I get this balanced? And the answer is 2 of these X's are going to have to move over here. So after diffusion occurs, I will be balanced on both sides. So an area of higher concentration, before we started this movement, there were 6 over here, and there were 2 over here. After the movement, all the body's trying to do is maintain homeostasis or balance. This is diffusion, the movement from an area of higher concentration to an area of lower concentration. Concentration gradient, the difference between high and low concentration. So the concentration gradient would have been within 2 on our previous example, because there were 6 over here and 2 over here. Well, that's a difference of 2. I need 2 to move this way to make 4 on both sides. Diffusion, concentration gradient. Osmosis, the diffusion of water. A solvent will move from a higher concentration to a lesser concentration. Occurs across a selectively permeable membrane that is freely permeable to water. Now, I'll show you this before we go into the next slide. Alright, bigger one of these. One, 2, 3, 4, 5, 6. 1, 2, 3, 4. Now, there's our solutes, or our solid substances. There are 6 over here and 4 over here. To make this balanced, how many's got to move to this side? And the answer is the gradient is 1. So if I moved 1 over here, now I got 5 over here. And I have 5 over here. Now, let's take this back the way it was before, 6 and 4. Alright, there's 6 over here now, and 4 over here. So when diffusion is occurring, it's wanting to move this direction. Now, let's give for an example, these circles are water. Now there's 4 circles of water over here, and I'm going to put 6 of them over here. 2, 4, 5, 6. Now, again, this is nothing more than the movement of from a higher concentration to a lower concentration. So whenever we take a look at the water's perspective, the water is going to want to move this way. Now, here's the reason why. One, there's more water on this side than there is on this side. So the water is going to move from an area of higher concentration to an area of lower concentration. And the solute, or the solid substances, are going to move from an area of higher concentration to an area of lower concentration. So all it's doing is balancing on both sides. Alright, so diffusion. Movement of a solute from an area of higher concentration to an area of lower concentration. Concentration gradient is the difference between the high and the low concentrations. Osmosis is pretty much the diffusion of water. It again will move from an area of higher concentration to an area of lower concentration, as long as the membrane is freely permeable to water. So this is diffusion, and diffusion does not require a semi-permeable membrane. So we drop this substance in here. This is like a colored sugar cube, and whenever it enters the water, the substance will diffuse until parts are equal in all parts of the water. Diffusion, area of higher concentration to an area of lower concentration. So it spread itself out. The cell membrane itself, some things are freely permeable. Example, water is freely permeable. These small molecules here, all of this stuff here, if the channel is large enough, it will easily allow these things to move freely inside and outside of the cell. However, things that are very, very large, proteins, this is a sugar, this is their example of a glucose molecule, this is an example of a very large protein molecule. There are not ports big enough to allow the movement through these open channels. So what we're going to see here is that there's going to be things like a gated channel that, when exposed to, like, say, insulin, will open up and allow the larger molecules like sugar into the cell. Okay, osmosis and osmotic pressure. Osmotic pressure, the force of water movement into the solution as a result of solute concentration. And all that is 1, 2, 3, 5, 6, 2, 3, 4, 5, 6, 2, 1, 2. Now, which way are the x's going to move? And the answer is the x's are going to move this way until they are balanced on both sides. Which way is the o's going to move? The answer is the o's are going to move this way until it's balanced on both sides. So what you get is the body is only trying to maintain homeostasis. The osmotic pressure is the force of water movement into that solution as a result of solute concentration. So the osmotic pressure gradient would be moving the water in this direction. When we talk about terms like isotonic, which is this one here, and isotonic solution is equal on both sides. And let me kind of explain that. Erase all this. So an isotonic solution is something that's balanced. So the substance that's inside of the cell is the same thickness as the actual solution itself. So there's no net movement. It's a very equal balance. Examples of isotonic solutions are things like normal saline and lactated ringers. The reason that we give people normal saline and lactated ringers is because whenever we put them into the vascular system, those solutions will have no net movement. So what it should do for us is give us an intravascular expansion or increase the patient's blood pressure. Hypotonic solutions, which is this one here, lessen the solution net movement into the cell. So in a hypotonic solution, the substance is thinner. So what it's going to do is the substance in the cell is a whole lot thicker than the substance that we just put in. Example of this would be like if we ran just water into the vessels or into the veins. This stuff here would have a gigantic net movement into the cell until the point where it would burst the cell in some case, which leads us to the next term, hemolysis, or bursting of a cell. So whenever something is hemolyzed or hemolysis occurs, the cell itself has been burst open. Hypertonic, example of hypertonic, the stuff inside the cell has thickness, but the hypertonic solution, example of this would be like mannitol, which is a protein, COOH, and then a sugar combined together, C6H12O6. So that's a gigantic molecule. So what do you think is thicker? Is it thicker inside the cell, or is it thicker from the solution standpoint? And the answer is this right here is much more thicker than what's in the cell. So we're going to get a movement of water towards the thickest material. I may have forgot one. And then crenation. Let me talk about this here for a second. I forgot one of the terms. So if we put this hypertonic solution into the extracellular environment, what we do is we get a movement of fluid outside the cell, and again until it balances, which if this stuff is really, really thick, we'll never get a balance. So what it'll do to the cell is it'll do something called crenation, which is essentially suck all the fluid out of it and super dehydrate the cell. And this is osmosis. This is figure 3.6 in your book. So as we look at this, if we want to pay attention, glucose molecules here and water molecules here. The little blue dots are the water molecules. The big purple dots are the glucose molecules. Now, this is a semi-permeable membrane right here in the middle. If we'll notice here that in step 1, there are more glucose molecules on this side than there are on this side. Here, there are more glucose molecules on the side of the check than there are on the side of the X. So what's going to occur is the water is going to move in this direction and try to balance the equation. Now, if this is semi-permeable, what it's going to do is it's not going to allow until the tenicities are equal, or the weight, or the molecular weight is equal. So, by the rushing of water in this direction, it decreases the volume on this side because of this semi-permeable membrane, and increases the volume on this side. Now, in this one, force is applied on this side that is actually going to equal the pressure. So what has just occurred is the force has been applied that is equal to the osmotic pressure gradient. Again, from everybody's perspective, the solutes are going to move from an area of higher concentration to an area of lower concentration. The water is going to move from an area of higher concentration to an area of lower concentration. In step 1's point of view, there was more water on this side than there was on this side with the glucose. So the water moved in this direction. If it's allowed to freely move and there's no pressure exerted against it, then this will cause an increase in volume on the side that there is fluid movement towards. If there is force applied, we will get movement until we equal the pressure that is applied upon it. So, osmotic flow across the cell membrane. So an isotonic solution, this is isotonic right here, an isotonic solution, there is no movement, net movement of fluid, so it's a balanced solution. Then in this picture here, the cells look fine. They're not dehydrating, they're not engorging, they're staying just like they are. In a hypotonic solution, which means that the substance that we just put in is thinner than what is currently in the cell. Everyone will take a look at the cell here. It is just about ready to burst. That's because the fluid was thinner in the extracellular area and rushed into the cell, because water is freely permeable until it has made the cell swell up, and it can burst the cell. The cell will pull water in until it is balanced on both sides. In the case here with the hypertonic solution, this is hypo, this is hyper, and this is going to be the thickest solution. So we have all of this solute out here in the extracellular environment. Now what this is going to do is this is thicker than the substance that's in the cell. So it is going to pull fluid from the intracellular compartment. And if everyone will take a note here, that those cells are crenated or they are super dehydrated. Clinical note, fluid movement. Discussion over this is going to be hyper, iso, and hypotonic solutions. So again, hypertonic solution. A lot of solute. This is hyper. The cell here, this is the cell membrane. It has about that much substance to it. So which is thicker? Is A or B thicker or have more weight? And the answer is A has more molecular weight. So what it's going to do is it's going to pull fluid from the intracellular compartment to the extracellular space. The next one we're going to talk about is isotonic. And this is the cell again. 1, 2, 3, 4. An isotonic solution has the same molecular weight as what we're introducing it into. An example of this is going to be 0.9% saline solution. It is an isotonic solution. So that means that it is going to be balanced. There is not going to be any net fluid movement whatsoever in an isotonic solution. Last but not least is going to be our hypotonic solution. And the hypotonic solution is way thin. As far as molecular weight goes. So since it's thin, which way is it going to, as far as the weight goes, which way is it going to move? And the answer is there's fluid everywhere else from an area of higher concentration to an area of lower concentration. The fluid is going to move inside the cell. Hyper, net, so let's just do this here. I'm going to erase this one more time and go do a quick review on these. There's my membrane, and this over here is all the cell. So in a hypertonic solution, this is going to be thicker fluid's going to move towards it. In an isotonic solution, very balanced. In a hypotonic solution, this is going to be thicker in the cell than it is in the extracellular compartment. So the hypotonic solution is going to move in that direction. So hyper towards the hypertonic solution, iso, balance, and then hypo into the cell. And this is the same thing that we just talked about. So if the hypertonic solution was out here, this is the thicker part, water moves out. Isotonic solution, nice and balanced. Hypotonic solution, this is going to be very, very thin out here. This stuff on the inside of the cell is going to be thicker. So water is going to move towards it from an area of higher concentration to an area of lower concentration to its perspective. Pardon. Filtration. Filtration. Passive process. Hydrostatic pressure forces water across the membrane. If solutes are small enough to fit through the pores of the membrane, solutes will be carried out of the cell. And pretty much what allows filtration to occur is blood pressure. So the hydrostatic pressure in our body is from blood pressure. Carrier-mediated transport. Carrier-mediated transport, membrane proteins bind specific ions or organic substrates to carry across the cell membrane. And this is going to allow membrane proteins to take certain substances across the cell membrane. Facilitated diffusion, and this is used in glucose and other proteins or amino acids. Passively transported across the cell membrane. No ATP use in facilitated diffusion. And then receptor sites. Molecules to be transported binds to a receptor site, the protein changes, and moves the molecule into the cell. Active transport. High bond of ATP provides energy to move needed ions or molecules across the cell membrane. So in ATP, if we have a cell membrane, this is to physically move it, we'll have a pump move part A into the cell, and then vice versa. Now we're going to see things like exchange pumps here later on, which is our third topic here. An example of an exchange pump is a sodium-potassium pump. Term 2, ion pump, actively transports ions across the membrane either into or out of the cell. And this is kind of setting us up for the next term, which is exchange pump. If the movement of ions are both in and out of the same pump, it is considered an exchange pump. And an example of an exchange pump is a sodium-potassium pump. Sodium-potassium pump homeostasis depends on the correct sodium and potassium concentrations. Sodium and potassium pump may use up to 40% of the ATP produced by the cell. So this uses a lot of energy, but in the sodium-potassium pump, we do things like generate neural impulses in our brain. We generate the ability to contract our muscles and so on. So big, gigantic exchange pump that the body uses, which is sodium-potassium pump. And this is an example or a picture of facilitated diffusion. Now, if ever want to remember back to the previous chapter, that stop sign looking thing is their representation of glucose. And what this did is insulin hit this receptor site and allowed, whenever the glucose got there, to be opened up and brought into the cell. Without the utilization of insulin, that carrier protein would have never enacted. And this is an example of the sodium-potassium pump. If you'll remember this seeding right now, that potassium is the most intracellular abundant ion, and sodium is the most abundant extracellular. Now, what occurs is in the sodium-potassium exchange pump, sodium moves in, potassium moves out. And this exchange changes the polarity on the cell and will allow it to shorten in the muscle cell's case. We'll talk about other aspects of the and use of this exchange pump in physiology as we continue. Tonicity of fluids. So this is IV fluid therapy, and this is a clinical note. Now, if everyone will come down here and kind of take a look for a second, as long as we're about 300, we have a substance that is considered to be balanced. There is no net movement of fluid. So Ringer's is an isotonic solution. It's about 309. And then let's see, there's our dextrose. Or, I'm sorry, there's our normal saline right here, which is about 308. So as long as we're right about 300, we're pretty good. We are considered to be isotonic. Closer we are to 300, the better. Now, we mixed all of our drugs in 5% dextrose and water. The reason that we mix everything in 5% dextrose and water is because it is slightly hypotonic. Now, what that does for us, if the cell is here and we introduce the drug into a hypotonic solution, it will want to go into the cell and also want to hit the receptor site inside the cell as well. So the little bit of a push that fluid has on that drug is why we choose a slightly hypotonic solution. If we'll look as this number gets larger, around 500, it is considered to be hypotonic. 10% dextrose injection. We only added 5% more dextrose, but the large molecule gave it more weight and made it hypertonic. So, as far as the hypertonic solution is concerned, cell, and we have a hypertonic solution here, the fluid is going to rush towards it because it weighs more than what's inside the cell. Hyper, hypo, and isotonic solutions. So, effects of IV fluids. And if everyone will notice here, this says hypertonic. So the thicker the substance gets, or the more hypertonic, the more fluid out of the cell that we get. And this will cause crenation or cause dehydration of the cell. So this one's crenated. If we have an isotonic solution, it's balanced. There's no net movement of fluid, so the cell is going to be normal. The more hypotonic that we become, the thinner that we get, the more that the fluid rushes into the cell because the stuff in the cell is thicker than what's on the outside of it. And if we even become more hypotonic, what this will cause is this will cause us to burst the cellular material, or cause the cell to burst from high osmotic pressure. Vesicular transport. Vesicular transport. Material movement in and out of the cells and vesicles. Endocytosis, endo and side. Packaging of extracellular material in a vesicle at the cell surface for import into the cell. We have receptor-mediated endocytosis, formation of small vesicles at the membrane surface to import selected substances in the cell. And as far as receptor-mediated endocytosis, an example of this would be iron formation or iron brought into the cell. These little things called ligands will form around the substance whenever it hits this specific receptor on the cell surface, and it will cause the cell wall itself to kind of envelop the substance that it needs to get inside the cell. Pinocytosis is another example of endocytosis, which is formation of a small vesicle filled with extracellular fluid. So I need a little bit of a drink, so the vesicle forms around the extracellular fluid and kind of brings it in. And then phagocytosis, which is cell eating, produces vesicles that contain solid objects that may be as large as the cell itself. And white blood cells are phagocytic. And what that means is, or macrophagic is another word for it, is they will eat or consume other cells that are not like itself. And then last but not least, we have exocytosis, which is pretty much the reversal of endocytosis. And a vesicle created inside the cell fuses with the cell membrane and discharges its contents into the extracellular environment. Exocytosis is something that we'll see in cells that specifically secrete things into the extracellular environment. And here we go is a little bit of an overview with this. These are the ligands that I was speaking of. These are receptor mediated. This will form a little pocket and then it'll kind of wall it off. And this is coated vesicle that allows a lysome, that's one of the organelles in the cell, to kind of break it down and take this part and do whatever with it, distribute it around the intercellular area. If exocytosis needs to be done, if this whole cell is secreting something, it's walled up on the inside and goes to the surface and will expel itself into the extracellular area. This is an image of phagocytic action. White blood cells are a good example of this. So it sees a foreign material that it doesn't like, it starts to wall it up until it encapsulates it, and once it encapsulates it, it puts it in a vesicle. The lysomes inside the cell attack it and break it down. It consumes what it needs or what it can consume, and then the parts that it can't, it expels into the extracellular environment. And this is the end of Chapter 3, Part 1. If you have any questions, feel free to contact me, 219-7613, or you can email me, smith@impsa.net. Again, if you will just call, I'll be happy to clear up any questions that you have. And thank you.